Okay. Great. So let's get started. We want all the time that we can get. So my name is Eytan Abraham. I'm the Chief Commercial and Technology Officer for Minaris, a cell and gene therapy focused CDMO. A privilege to moderate this panel. I think we're all aware of the very significant opportunities, but also challenges in terms of developing, launching, and treating patients with regenerative medicine, type therapies that are cell-based, combination products that are cell-based. These are significant challenges. They've been sort of the holy grail of the cell therapy industry for many years. And I think we're getting close. We're getting closer and closer to launching these commercially. So we have three extraordinary panelists. And they represent different approaches, different technologies, different methods to make this work and to get to patients and treat patients. So I think it's going to be a very exciting, interesting session. What we're going to do is we're going to do a brief 10-minute presentation for each of the panelists, and then we'll all get on stage and we'll have a discussion. So looking forward to it. Please, let's get started.
I assume everybody of you has interacted with a diabetic patient already. If not everybody, almost everybody. And you have been seeing how they are suffering from daily insulin injections, from prick tests, today with pumps, urine tests. And I think if you're not a diabetic patient, you can only imagine what it takes, what kind of endurance it takes to treat this disease, to be maintained at good glucose and insulin levels. What we are presenting to you today, three different approaches, will definitely not be the final, the best product to treat diabetes. But what we are presenting to you is essentially solutions that will finally deliver all of our objective, which is about to cure diabetes. Because this is what all unites us who are going up on stage today. With this introduction, welcome to this panel. My name is Bernd. It's a quite unique situation for me because I am presenting both Sernova and Evotec. I'm board member of Sernova Biotherapeutics, and at the same time, my professional life, I'm heading cell therapy business at Evotec. The collaboration between Sernova and Evotec is about delivering a fully retrievable cell therapy, which provides functional cure. And it's made out of two elements. That's a so-called cell pouch, a biohybrid organ, which I will show you. And it's made out of islet-like clusters that are derived from iPSC. What is important from our perspective is that any treatment, any cell therapy treatment, needs to be retrievable when we are approaching the final cure for diabetic patients. Why? Because this enhances the safety. It decreases the long-term risk of the patient, because the question is, how do you extract the cells in case something goes wrong, they become cancerogenic or whatsoever. And there is also an impact on regulatory timelines and risks, because you're working with stem cell-derived cells. In the future, we will be working with genetically modified cells. So we believe it's in the best interest also of the patient to have a system delivering the cells that is retrievable. The cell pouch that is being developed by Sernova, you can see a schematic presentation on the right-hand side. It's an 8 or 10 chamber device. The important thing is this device is pre-implanted into the patient. So 4 to 6 weeks before the patient receives the cell load, this device is implanted, and it integrated and vascularizes into the tissue. As you can see, it's implanted on the left bottom, essentially on the abdominal muscle. So this is not meaning it's a 4-pack or a 6-pack. You don't really feel that device because it's so soft that it's not really visible on the abdominal muscles. And once the device is vascularized, so essentially after 4 days, there is another surgical procedure where the plastic rods that are within the chambers are removed, and then the cells are filled into the device. The current clinical trial, it's a phase 1/2 trial with a cell pouch, is currently using cadaveric islets that are donated. And so what is important to mention is that the device developed by Sernova has shown long-term safety and tolerability, which really makes it very, very different and unique from other devices that have been used in the past. What does it mean? This device has been in a patient for more than 5 years, and in total more than 33 years of accumulated lifetime in patients, and we have seen no serious adverse events, and we have seen no fibrotic tissue rejection at all. The patients that entered these clinical trials have all been C-peptide naive, so they don't have any C-peptide levels. And what we have been observing is that all patients that received the immunosuppression regimen and followed the immunosuppression regimen diligently received C-peptide levels and have been insulin independent following a single low-dose interportal transplant after cell pouch implantation. So overall, what we can say is that the cell pouch supports really long-term cell engraftment after 5 years. This is how it looks like. To the left side, you can see the cell pouch before implantation. To the right-hand side, you see the pouch after it has been explanted more than 50 months within the patient. And as you can see, it really looks like a perfect organ. There's no tissue problems. There is no fibrotic areas around this biohybrid organ. And this is why we believe that Sernova's cell pouch is one of the key elements in the future to deliver cells to patients and to cure patients. How does it look like? What you can see here is the extracted cell pouch, and there is one histochemistry cut, so to say, across all 8 chambers. And what you can see at the bottom, and I'm not sure it's nicely visible because the room is so light, is staining for insulin and von Willebrand factor. So you can really nicely see that within the cell pouch chambers, we see abundant production of insulin and also blood vessels. This is not only true for blood vessels and insulin. What is important is the composition of the islet-like cluster. So you need beta cells, you need alpha cells for glucagon, somatostatin you need, and so this is all visible and nicely produced from within the cell pouch. And please remember, what you are seeing here are cells that have been donated. So cadaveric islet cells that have been donated and that have been within a patient for 5 years. And that's quite unique. So the intent is to replace this with islet-like clusters, iPSC derived. And before going into the manufacturing of this, what you can see here in the graph, that is an animal model where you can see the glucose level was essentially destroyed with STZ. And then these mice were implanted with islet-like clusters derived from iPSC. We are actually targeting an immature beta cell stage for implantation. So there is a final maturation step of these islet-like clusters within the animals. And that's nicely seen. You can see after around about 100 days, we are receiving a stable, very nice, stable level of glucose in the mice. What is important is that human islets run below the glucose set points of mice, which is the dotted black line. And as you can see, we are really achieving this very nicely, showing full functionality of the human islets within the mouse. And you can also see that C-peptide and tolerance testing work perfectly. With this starting point, we invested massively into the manufacturing in our GMP facility. It's a 2-module process. First module is essentially to manufacture the cells, to harvest them then at late stage and freeze them so that they can be shipped globally. And then in a regional setup, process module 2 will be implemented, meaning the cells will be thawed. QC for administration, filling, and a little bit of final maturation is done, and then shipment to the site as a living cell culture. When we use the cell pouch from Sernova in mice, obviously we don't use the one that is in patient, but a tiny one. You can see to the left-hand side, that was one of our first experiments that we are also achieving very nicely here, the human set point within the mouse. And also C-peptide levels, which is the panel to the upper right, is really very nicely equivalent to the same dose of 4,000 IEQs human islets when administered to a mouse. And also glucose tolerance testing is equal to the human islets that are donated. Finally, this is not just a one-shot experiment. We have several studies that we have been doing, and when we are comparing our working horse device of the past, which is the usual Terasite device being used in the mice, with the cell pouch, we can really see in these pictures where C-peptide levels are shown that we are able to really generate appropriate and good C-peptides level after 6 weeks of transplantation into the mice, which is exactly the time period when we see full maturation. And you can also see to the far right, the third study, that this is the dose-dependent achievement depending on the level of dose that we are administering to the mice. So overall, this is a full and sustained glucose normalization that we can achieve across all dose levels. To sum up the status quo where we are today, we are advancing this into a first-in-human study. We have the long-term safety and tolerability in patients with type 1 diabetes for the cell pouch. We have really seen that cell functions can be restored for 5 years within the cell pouch. We are now generating the islet-like clusters from iPSC, and we know that they are absolutely identical to the human islet-like clusters. We have a modular GMP process, and it's now on us to combine all of this to advance this combination of technologies into a first-in-human study that we are targeting in the second half of 2026. So that, in a nutshell, is one of the technologies which we believe will make a change. And the next speaker, Vlad, will be coming on stage and showing you a very different approach, but again, with the same objective. Thank you.
Thank you very much for the invitation and be part of this really interesting session. Laverock Therapeutics is an early stage startup biotech, and we've started with a new gene silencing technology. We're now established this platform technology, and we're looking to develop therapeutic products based on this new technology. So I'm going to spend half of my talk describing the platform technology and then how we think this could be applied to, and how we're looking to apply it to develop allogeneic cell therapies for type 1 diabetes. So our technology is a new way of leveraging the RNAi, and you'll be familiar with siRNA and shRNA approaches. These are introducing either synthetic oligos or transgene precursors of hairpin precursors of shRNAs. Instead of doing that, we're using the cell's own RNAi genes, which are these non-coding RNA genes called microRNAs. There's hundreds of these, thousands actually annotated around our genome, and they're the reason the RNAi pathway exists. The RNAi machinery is there to mediate its silencing function. So they work, by and large, very similar in equivalent manner to siRNAs, only that they're already encoded in the genome and they have their own program of expression. So we identify which microRNAs are expressed in the cell types of interest, and then we introduce very specific edits in their sequence to redirect their silencing activity towards our desired target. And the main advantage of this is, so we're not introducing any exogenous regulatory elements, and the main advantage of this, it's a unique combination of features that improves on existing gene silencing technology and gene knockout approaches. The main one being the fact that you can program when gene silencing activity comes on. So on the bottom right, we're showing essentially that you can program when your target is silenced, so it's not constitutive, which would be the case if you apply a gene knockout approach. But if you take the example of an iPS cells, you can engineer it so that this IPS line is otherwise identical to a wild-type line, but then it will specifically silence your desired target in your target cell type, so the silencing activity can be programmed to come on at the right time. So how does this work? On the bottom left here is an overview of what microRNA expression looks like in different tissues. On the Y, you have the microRNAs annotated in the genome, and the X-axis, you have the different tissues. So you can immediately see that there's hundreds of, thousands of these microRNAs that have very distinct programs of expression. Some of them are more broadly expressed across tissues, but some of them can be expressed in a very cell type specific manner. So because we are essentially using these microRNA genes as vectors of expression for these new silencing RNAs that we design, we basically borrow these programs of expression. We borrow the program of the expression, but also their abundance. So this means that you can, with this technology, you have instant access to hundreds of different expression patterns that you can use to program when the silencing activity of your target comes on. And this is what we illustrate here with these IPS lines that we generate, in which we specifically edit a microRNA, which is silent in iPS cells, but then is specifically induced in the myeloid lineage. So you can see in the top right, the expression of the silencing RNA is inexistent in iPS cells, but then it gets strongly induced in monocytes, and then its expression increases in macrophages. So on the bottom right, you can see that the target, it's not silenced, so it remains the same in iPS cells, but then gets very effectively silenced in these differentiated cells. So in this way, you can achieve this cell type specific gene silencing. This technology also allows you to tune the expression of your target. So as I was saying, it's not just the tissue-specific patterns, but also the different abundances of these microRNAs. So by leveraging these microRNA loci as vectors of expression, you borrow not just the tissue-specific patterns, but also the abundance of these microRNAs. And because they are very reproducible, they're also very predictable. So in this case, we can generate lines on the top left where we show different levels of the target modulation. So in this case, these are components of the MHC1 complex, which is directly relevant to generating hypoimmune cells. So by reducing the expression of MHC1, you can make cells that are hypoimmune, so you can evade the rejection by the patient's immune system. And you can see that the expression of the target is very homogeneous within this population. So this is something that it's impossible to achieve with gene knockout or even with transgenic shRNA. It is also very highly specific, and this is another feature of a consequence of driving expression of these silencing RNA from endogenous microRNA loci, because they're never overexpressed. They're always expressed within the physiological boundaries of what a normal microRNA is expressed at. So you can see that in this case, the only gene that is differentially expressed is our specific target. This allows us to generate cells in which we can selectively silence, for example, HLA genes. So we can generate these cells in which only the classical and polymorphic HLA genes are silenced, but not HLA-E, which is an important factor in maintaining tolerance, especially in inflammatory conditions. Finally, it also allows you to multiplex silencing. So in this case, by editing a single locus, you can express multiple silencing RNA. So this addresses the complexity of genome engineering when you need to silence multiple factors, especially in the case of making hypoimmune cells. So how are we looking to deploy this technology in the treatment of type 1 diabetes? And this is essentially generating pancreatic islet cells from iPS cells. So as Bernd mentioned, this is already an established and FDA-approved treatment. And the main problems that these approaches face based on donor-derived approaches is they're really largely two categories. One is the limited cell source, and this is where we represent essentially the disparity between supply and demand in this case. which is 3 orders of magnitude between the availability of these pancreatic islet donor-derived pancreatic islets and the patient demand. And the second part is administering them, so putting them into a patient faces 2 main challenges around graft rejection. One is the alloreactivity, because it's virtually impossible to HLA match between donors and patients. And the other one is the autoreactivity. So these patients have lost their beta cells because of an autoimmune response, which is still there. It doesn't go away, so you have to deal with that as well. So these are addressed in a few different ways, and for the limited cell source, the stem cell-derived or iPS-derived pseudo-islets, it solves that problem because you can generate really large quantities of it. There are also more experimental approaches to transdifferentiate alpha to beta cells. And in graft rejection, you have a few options: patient immunosuppression, so medical immunosuppression, encapsulating them in immunoprotective devices, but also engineering hypoimmunogenic cells. So engineering the iPS-derived pseudo-islets to evade the patient's immune system. So these are still facing significant hurdles. And I'm going to mention mainly the 2 ones. In the stem cell-derived pseudo-islets is still inconsistency of manufacturing, so batch-to-batch variability, but also the existence of contaminating undesired cell types. And in the field of graft rejection, so engineering these cells requires complex cell engineering, so you need to be able to silence multiple factors in these cells, so not targeting a single pathway, just not sufficient. And also, once you make these cells hypoimmune, then you increase the risk that if these cells stray off, they lose cell identity and they transform, they can generate these immune-evasive malignancies. So this is where we're looking to apply our technology to provide some solutions to these hurdles. So this is where we represent in rough manner the trajectory of iPS differentiation to the pseudo-islets. And even with the best protocols today, you can still get some contaminant cell lines, some contaminant cell types. So an example would be enterochromaffin cells that derive from the same common progenitor as beta cells. So the idea with our technology is that we can map microRNAs that are specific to these intermediate progenitors, which are the stage gates that decide in which direction these cells will differentiate. And then we can redirect those microRNAs towards key lineage decision factors so that we can block the differentiation of these contaminating cell types. So the idea here is to increase the purity of the final product in the pseudo-islets. And this is important because the islets differentiate into cell clusters that form gap junctions, so they're not really amenable to breaking down, enriching, and then reaggregating. The other application is in graft rejection, and I'm not going to mention targets because there's loads of different approaches here. The main thing that I want to discuss is that most approaches that are out there, they do generate this constitutively hypoimmune cells. So the genetic engineering that is applied means that the iPS cells, intermediate progenitor, and the final products are all hypoimmune. So we can, and this means that, contaminating iPS cells or undifferentiating iPS cells that can form teratomas, but also dedifferentiating cells that lose cell identity, they can generate these immune-evasive malignancies. So with our technology, what we're looking to do is to engineer iPS cells that so that these immunogenicity factors are only silenced when the cells are differentiated into the final product. So we can redirect these microRNAs that are specific to mature islet cells so that these immunogenicity factors are specifically silenced in the final product, so that if these contaminating iPS cells or dedifferentiated cells exist, they're still fully immunogenic, so they can be rejected by the host. Also, the modularity of the technology, again, at the moment, the combination of targets that we're addressing, because this field is really in its infancy, it's very likely that it will require multiple rounds of iteration to find the right combination of targets to silence so that we can address both cellular immunity but also antibody-driven immunity. So with our technology, would allow, due to its modularity, once you've identified these microRNAs and you've established them as the right loci to drive the silencing RNAs, then new targets that come from the R&D or other collaborations, they can be included in this multiplex design in a straightforward manner. So then you can swap in or add new silencing RNAs to address new pathways. So finally, this would be the blueprint of this product that we're aiming for. These will be IPS-derived, and all the programming will be done at the IPS stage, allowing us to do clonal selection and extensive QC. So then, as the cells differentiate, different microRNAs at different states in the differentiation would activate silencing specific to those progenitors to block differentiation of unwanted contaminated cells. And finally, in the fully mature differentiated cell products, you would have expression of multiple silencing RNAs that block multiple pathways that mediate immune rejection. So thank you very much for your attention, and I think next we'll hear from Harald, who's going to tell us more about immune-evasive encapsulation devices.
It's a privilege to follow in the footsteps of Bernd and Vlad. As Bernd and Eytan mentioned, the purpose of this Science Slam is to introduce you and us to a number of complementary technologies that hopefully will bring us closer to our goal of treating type 1 diabetic patients. My name is Harald Stover. I'm the CEO and CSO of Allarta. If I mention the name, maybe that'll move the slides to the Allarta slides. We are a company that's rooted in synthetic polymer chemistry. We've been working on immune-evasive hydrogels for a couple of decades, and over the last couple of years, we've translated this into a therapeutic approach, as was mentioned, encapsulation of beta cells. I am clicking, but nothing moves. Ah, awesome. Okay, good. That's where I wanted to be. So we are based in Ontario, Canada, just outside of Toronto. And our technology is based on an immune-evasive materials that we call hydrogels. The thesis is that no matter whether you use hypoimmune cells, donor-derived islets, stem cell-derived beta cells, we believe the cells can benefit from being inserted into an immunoprotective and immune-evasive hydrogel, shown here by the blue network. The green are the cells. The idea is that these materials are permeable enough to allow indiffusion of glucose, oxygen, other nutrients, and of course allow for outdiffusion of insulin and any other therapeutic proteins that might want to be expressed, but are able to deflect cellular and molecular components of the immune system. So key to our approach is synthetic hydrogel networks. They're covalently cross-linked for persistence. They have lasted in vivo for up to a year so far, the lifetime of a mouse, in vitro much longer. They're designed to be immunoprotective, to be retrievable, and tunable for the reasons the previous speakers have indicated. This video illustrates a little bit how we combine our polymeric gel formers, these are synthetic cross-linkable immunoevasive materials, with donor islets or stem cell-derived beta cells into a viscous liquid that can self-cross-link in capsular form or other shapes. We can coat it with different materials to tune permeability. And we work on different shape factors that all have in common that the encapsulated cells are permanently immobilized. They cannot escape, nor can immune cells diffuse in, and we have shown that with histology. But the network is permeable enough, as I mentioned, to allow nutrient and oxygen access. None of the cells is further than about 200, 300 microns from the interface with the tissue. So overall, that technology, that composition applies to all shape factors. We think of it as a platform for immune-privileged delivery of cells. And we think, in the spirit of this science slam, that it takes a village to build a therapy. We can provide physical encapsulation. We can provide a standoff from the immune cells and the innate immune system of the host. Of course, we are still communicating. There are still damp factors coming out. But so far, it seems that they are diluted enough in the animal models we've chosen to not affect the outcome. But soluble peptides that are being produced is still a challenge for us. Surface markers, perhaps less, because we don't have cell-cell contact. Just to illustrate some of the attributes of these materials, on the left you see a live sphere being compressed against a point. Vacuum capillary, you see the deformation. This is underwater. They express some media and then they jump back to the original form. Lifelines can be extruded into a gelling bath. You see human donor islets centered in the string, and we can make multi-meters within about 2 or 3 seconds. And on the right-hand side, you see lifelines basically 3D printed with a very simple off-the-shelf 3D printer into a rat model device, 3 layers, the cells inside the gel strings. There's lots of space for vascularization. And again, all of these devices can be post-coated for immune evasion, for control of molecular weight, such as exclusion of IgGs, which is one of the key issues in type 1, in people with type 1 diabetes. In our field, of course, we use immunocompetent animals as preclinical proofs of concept. These are the black 6 mice that we render diabetic with STZ. You can see the high blood sugar at the beginning, 400, and after transplantation of, in our case, a fairly marginal load of rat donor islets, about 1,000 IEQ in the peritoneal cavity, blood sugar is controlled at a reasonable level and persists for almost a year. On the top right, you see the same with STZ diabetic Vista rat. That's an allogeneic transplant, rat islets into rats. These are outbred rats, so it's a valid model. And on the bottom left, of course, we go to one of the ultimate therapeutic payloads, which is, in this example, human donor islets. We transplant again about 1,000 human donor islets, or IEQs, islet equivalents, into diabetic black 6 mice, immunocompetent mice. The red line shows the blood sugar control out to almost 50 days, while the gray line shows what happens when you implant free islets. They do not affect any blood sugar control. On the bottom right, we show early experiments with diabetic and healthy pigs, both Göttingen and mini pigs and Yorkshire pigs, where we've implanted, again, marginal human donor islets into the peritoneal cavity. We see blood sugar control, as well as C-peptide and functional live islets, certainly 2 to 3 weeks out and probably beyond. The live rafts are our most exciting technology long-term because they represent an implantable and retrievable device. You know where the surgeon puts it, whether it's 1 device or 4 devices, similar placement to where Sernova puts their devices, lower abdominal region. And shown here are some data with mesenchymal stromal cells. This is one way to, there's one cell line we can reproduce in the lab very easily, and it has a number of interesting applications. You see high cell viability on day 0, day 1, day 3, pre-implant, and on the right-hand side after 4 weeks in an immunocompetent rat. And again, the open space between the strands gets vascularized, and the cells show good viability. In addition to dealing with the implantation and immune evasion within the patient, because we are polymer scientists at heart, we are always interested in addressing other logistical challenges in this field. One of them, of course, is scale up for stem cell expansion and perhaps differentiation. These cross-linked capsules on the top left were seeded with a small amount of iPSCs, which were then grown up to fill the capsules, but because of the cross-linked nature of the shell, the cells do not escape. They are trapped inside a straitjacket, if you like. And on the top right, we show an example of a collaboration with Pancella at the time. It's now part of Pluristics. These are iPSCs that include a suicide gene that can be triggered after the cells have expanded to a fairly large cluster by administration of ganciclovir. And you can see with a red line on the graph at the bottom that the cells can be ablated rather efficiently. So capsules, as you can imagine, don't interfere with the metabolic exchange or with any other triggered interference that you may need to do. Excuse me. Oh, here we go. Another logistics challenge is cryoprotection of cells. We've been using DMSO for many years. At Allarta, we're trying to replace this with the blue microgels that we can conformally arrange around cells and small cell clusters. They act as polymeric DMSO, if you like. They prevent ice crystal formation. They do dehydrate the cells to enable the cytosol internal cryoprotection that is necessary during cryogenic storage at -80 or liquid nitrogen temperature. The cells come out and they can be washed clear of the microgels. They can also be degraded. So this is an approach that we think has multiple applications in the value chain from cell production into the patient, potentially including cell storage in the basement of the hospital, or even room temperature storage, 4-degree storage during transportation. The left image shows human iPSCs that are stored in these transparent jammed gels. It looks like cars parked in a parking garage at different levels, so they are fully metabolically connected by diffusion. They don't aggregate, and we have seen many benefits in using these matrices to handle some of our cell clusters. We can also use them for cryoprotection of small iPSC clusters. On the right-hand side, this is not a diabetes application. We can cryoprotect brain endothelial cells and INK cells with better post-thaw viability than with the current standard of care 10% DMSO solutions. So we think it's another example of it takes a lot of different technologies to scale cell therapy to a safe level. One last example of the cryogels, shown here again with MSCs. The panel on the top simply shows good survival in 0 and 2% DMSO inside the microgels. And the scatter plot shows that you want to be in the top right corner where where our formulations are competitive with 10% DMSO at lower toxicity, which is important. DMSO is not good for cells, caregivers, or patients. We are an integrated company. We thought we needed only 5 people, but we are now at 35 and still growing. We are based in polymer science, and we follow up with engineering, cell biology, and our own preclinical team. And of course, the technology has application in many other indications. With that, I'll wrap up and ask Eytan to come and assemble us on the stage. Thank you.
Great, thank you. Really very interesting talks and different approaches. Maybe I'll start with asking a question, which is thinking about the end in mind, meaning treating potentially hundreds of thousands of patients with these therapies. Maybe each of you can comment on what, for your specific approach, you think the biggest challenge is in that regard. Who would like to start? Let's start.
I think, obviously, I think the efficiency of differentiation, that is very important. There's, even at a current sort of research scale, these are expensive to run. and can take quite a long time. So making sure that even sort of marginal gains in the efficiency and purity of the samples can actually make a big difference at a large scale. So that's why we thought that this application of our technology to maybe skew differentiation so that you get the optimal maybe ratio of cells, of alpha, beta, delta cells, but also get rid of impurities so you get maximal activity from what you have, would really help with this driving down the costs of the final product.
Fully in what you said, differentiation, the manufacturing is key also in terms of cost of goods. I think also what we need to think about is longevity of the approach. So if we treat a patient with cells, and whether they are encapsulated, whether they are in a carrier, whatever format, because of the cost for this treatment, which is in the several $100,000 dollars, this needs to be a sustainable treatment. So we are not allowed to do this twice a year from a cost effectiveness perspective. So what we need to target, and this is, I think, why we will be seeing in the future iterations of products as they evolve over time, is, in my opinion, that at least this needs to be sustainable for 5 or even more years. So that really the quality of life of the patient is not impacted any longer, and we have a good equation also for calculating the return for the insurance companies.
Yeah, fully agree with that. I think Bernd and I would agree the manufacturing of the devices, in our case hydrogels, is not a big problem. These materials can be scaled. Hydrogels in particular are mainly consisting of water. The longevity is an issue. With human donor islets, that is easy to address. They have worked in the Edmonton protocol for a decade and longer. It's always a question how long they will function our devices. We look at 3 to 5 years as well. I think we see scale up in 2 stages. We do think that human donor islets have a role to play, at least at the beginning. We think they are insufficient in numbers, but they are underutilized as well, and they are fully safe. And there will always be a population of people that will say, we'll wait 20 years until we have our kids implanted with an unknown new material. Donor-derived islets have been around for a while. So we think that may well be a starting point, but stem cell-derived cells, hypoimmune modified cells are an engineered product, much easier ultimately to manufacture and use.
Okay, so scalability of manufacturing, increased efficiencies in terms of purity and the cell characteristics and phenotype. So those definitely make sense. Bernd, you mentioned that currently with the pouch using cadaveric cells, it seems to work quite well. So that begs the question, why do we need to do iPSC-based? I mean, one of the elements is lack of supply, but is that really the issue, or is there something else?
No, I mean, the reality of cadaveric islets is, on average, you need 5 donors to treat 1 patient. So you have a massive, massive logistical challenge here. Secondly, these patients need to be under immunosuppression, right? Unless it's encapsulated as an additional step or whatsoever. So personally, I think from a process perspective, from a logistical perspective, because you need 5 to 1 equation, whereas this is a niche opportunity these days, it is not a viable product for this large type 1 diabetes or even type 2 diabetes market on the long run. So simply because of the size of the market, this is not the solution that is viable. So in the end, I think what we need to aim for is a product that is implanted into the patient in a one-time procedure that is durable over a long time and that does not require any immunosuppression. And I think if we are looking for that, if that is our target profile, the cadaveric islets are far away from that target profile, no matter whether you encapsulate them or not.
So, but that's an interesting observation. So if you encapsulate them, are they not immune privileged?
Of course, they are immune privileged, but it remains that you will never have a pharmaceutical product available, right? As I said, they are donated on average from 5 patients. And so you have a 5-time background that is not belonging to your physiology. So this makes it complicated again. Process-wise, you have 5 times QC checks, etc. So the whole setup is reasonably complicated. Of course, if everything would be perfect, that would be nice, but nothing is perfect. So what I think what we need to aim for is, and I think that's also a lesson learned from the autologous products that we are seeing, where we are seeing a tendency towards decentralization. Pharma companies don't want to sell a process, they want to sell a product. So if we want to come to a really cure for patients, I think we need to deliver what is compatible with our industry. We have to have an off-the-shelf product. And if we think about an off-the-shelf product, we need something that has clear specifications. And so this brings us to the point that we need to manufacture the cells in a reliable and reproducible manner at high volume so that we can supply this whole demanding patients across the planet.
Yeah, music to my ears. Yes, absolutely. So, yes, please.
Yeah, no, I mean, I totally agree. I think there's, so currently so initially when this approach was you know, set up without encapsulation devices, right? So patients needed to be heavily immunosuppressed. And that actually limits the accessibility of this to patients because the risk from immunosuppression is really high. I mean, these patients become very susceptible to risk infections, you know, reactivation of dormant viruses and malignancies. So for that reason, I mean, there is, for example, in the UK, you can get this on the NHS, but the condition is that you have to have 2 hypoglycemic events, severe hypoglycemic events over the course of 2 years. So that it's about 20 to 25% of the population. So that limits massively the number of people that can have access to that. And the problem of availability there is just insurmountable. So the slide I had up before, I mean, it was a cartoon, but it's based on real numbers I got from the US statistics. So there's about 1.5 million patients with type 1 diabetes. And there's about 1,300 of these pancreata available every year. So as Bernd said, this process of actually extracting islets is very inefficient, so you don't have 1 donor per patient, but multiple donors per patient. So it's just, the difference is just insurmountable without these stem cell derived.
Great. So I want to switch a little bit and talk about how you look at this or analyze it from the perspective of that this is a therapy that there are current solutions that would compete with it, right, commercially. Right, so there have obviously been advances in terms of insulin and insulin pumps and patches and other types of delivery to manage the indication. And how do you look at this from that perspective? Is it a question of clinical efficacy, ease of use for the patient, cost per patient? What are the main drivers for needing a different solution? Maybe you want to start, yes?
Yeah, that's a very interesting point. I was at the Laverock Symposium in Los Angeles last year, organized by Fuad Kandil, who, by the way, is now transplanting or curing type 1 diabetics with 1 donor loads of islets using certain peptides to boost their function. So that ratio has moved from 5:1 to 3:1 to 1:1. But it is still ultimately, just to follow up on the last point, donor islets are not an engineered product, and only 50% of pancreata can be transferred into usable islets. But on the comparison with closed-loop systems, Los Angeles County, which is all of the city of Los Angeles, provides free closed-loop systems to anybody that needs one. But the uptake isn't as big as expected. There are a lot of people that say, I do not want to have a device on me that I have to look after. I would rather have, even now, an islet transplant with immune suppression. I just have to take a few pills. So we think the future is, I think we're all aligned on that, definitely on cell therapy. a one and done, at least for 3 to 5 years.
Understood. So switching over maybe to safety, I mean, I think, Bernd, you mentioned that the device should be retractable for reasons of safety. And I'm wondering what safety specifically events are we concerned about in terms of needing to retract the device, or conversely in terms of needed to have a kill switch, right, to eliminate the cells. What are the main concerns?
So as I said, I'm a true believer that we will be seeing iterations of products, right? And I think the final ultimate goal is something gene edited that you get infused into wherever and you're done, right? Just imagine going to the doctors, you get it into a syringe, off it goes. That is the ultimate goal. I think until we get to that ultimate goal, we need to think in iterations. And today's starting point, in particular when we talk to regulatory agencies, essentially, to be honest, we don't have a bandwidth of experience of using iPSC-derived products or ESC-derived products in patients. We don't have that, right? So it's a legitimate request by the agencies to protect the safety of the patient despite being able to cure a disease. On the other hand, if we think about diabetes, this is nothing that, you know, this is not fatal in the next second. So it's something that is manageable over time, and the patient survives for a long time. So we need to make sure that we deliver something to the patient that is safe, and in that regard, having no experience with iPSC-derived products so far in patients at a large scale, we truly believe that the retrievability is a key element. You know, you don't know what happens, and just think about something is going wrong. You have iPSC-derived cells that become tumorigenic, and even if you have a kill switch, you know, you can run some math. If a kill switch has 90% efficacy and you administer 1 billion cells, you have a problem, right? So if the kill switch is not fully working. And so we really believe that in the first iterations of the product, no matter whether they are non-edited or even the first generations of edited product, simply to protect the patient and to ensure that we can really take off the cells immediately, which means patient goes on insulin again, and that's what we are seeing with the Sernova product. So if you retrieve the device with the cadaveric islets, the patient goes on insulin again and it's working. There is no ambiguity or no concern about this. And this is why we believe that retrievability for us in the first instance is really a key element to develop a safe product and to develop along the pathway of coming to the final solution in a meaningful way.
Understood. And Harald, maybe you can talk a little bit about the kill switch approach and what that entails, and to the point made, is it 100% effective or not necessarily 100% effective?
They are, what I showed was an example, and I understand it is highly effective. There are a number of other techniques, similar kill switches available. I would not rely on them entirely, as Bernd said. The risk is just too high. They may work, 100% in the Petri dish, but I don't think even that can be translated into the person. So absolutely physical retrievability at this early stage, perhaps for the next 20 years until we have longevity in patients, is a reasonable ask by the regulatory agencies.
Okay, and with regards to Vlad, to your approach, what is the regulatory, what are the thoughts from regulatory agencies or the concerns?
Yeah, I mean, this is still at an early stage. So I think that, I mean, if you look at the primary islets, donor-derived islets, so they are administered without encapsulation, right? So they are infused in the portal vein, they get trapped in the capillaries of the liver, and they kind of function as this sort of decentralized pancreas. So ideally, you would want to reproduce that or something like that. I think having the ability, but I think, with iPS-derived pseudo-islets, that is probably quite a leap of faith because there's a few different challenges or a few different risks, right? It's not just only one. the fact that these are IPS derived, so there are concerns around whether the differentiated cells have a robust cell identity program as the primary cells have, and they might be more prone to dedifferentiation. And on top of that, if we're engineering them to make them hypoimmune, then that compounds that problem. So the way we think about it is that in order to, for the regulator to make that sort of leap of faith, you will have to take this approach as belt and braces. And maybe an encapsulation device might be an intermediate step. But if you wanted to go beyond that, then I think you'll need multiple pathways to control these different risk factors. Our approach of making conditionally hypoimmune cells so that only the fully differentiated cells then go on and silence these immunogenic factors. We can also connect that with other pathways so that we can block, for example, cell proliferation, so that if the cells do undergo this transformation process, which is still a bit of a black box, there's more than one sort of mechanism that stops them from doing that. So I think that, maybe a single sort of mechanism as a sort of kill switch might not be sufficient, but you might, the same way we're addressing multiple immunogenicity pathways, we also might have to have multiple sort of safety mechanisms as well.
Great. So maybe last question, we're about at time. Maybe last question, I don't think now it's more correct. So where do you think we'll be in each of you in 5 years from now? I mean, how are we going to have a commercial product? How close will we be to a commercial product? How much will we have to iterate on these concepts to get to a point where this is really an effective therapy that can be used to treat many patients? Harald, maybe you want to start?
Yeah, this is a dangerous question. Where do you want to be in 5 years? JDRF has been promising a cure in 5 years for the last couple of decades. So I don't want to predict, but having said that, of course, we do want to push to the clinic. Patient advocates in type 1 diabetes are very adamant that new therapies are needed. They are willing to take more risks than the regulatory authorities. We think, again, leading with cadaveric islets, encapsulation can be ready for the clinic in, I'll say, 2 to 3 years, because the materials are harmless. For stem cells, we already have clinical trials that have been ongoing, but I'll leave that to Bernd and Vlad.
No, I think in 5 years, we will have more than one product with proof of concept in humans. I think the major difference to our environment, because if you look at the environment here, the type of companies we are sitting here, I truly believe in 5 years we will be having at least 2 big pharma companies entering the field, which is absolutely mandatory, because I believe that the companies that are currently entertaining these different technologies, and there are others also here at the conference, we don't have the muscles to put that together. So I'm convinced that on the backbone of the combination of what we do these days, we will be able to generate sufficient evidence for big pharma companies to join the show, to take the money and fund a cell therapeutic approach for the treatment of diabetes. And again, that doesn't mean we will have found the final product, but we will be a significantly step towards a registration trial. That I'm 100% convinced.
Yeah, I think, looking at where Vertex is, for example, so they have acquired some of the major players in the field. So with Therapeutics, via Seitan, they've done a couple of clinical trials with their encapsulation devices. And recently they've announced that they're abandoning the sort of encapsulated approach, but they're still pursuing this an engineered cell therapy approach in which they're trying to evade the immune system. So that is going to go into a clinical trial, and we're definitely going to have an answer in a 5-year time. And definitely, Bernd, good luck to you guys as well with your trial. I think it's important to have a proof of concept with positive data, but I totally agree that that's just going to be a first step. And then, but that will definitely provide more wind in the sails of companies that, you know, develop these different approaches, and perhaps through collaborative agreements, then we can develop the sort of follow-on generations.
Excellent. Okay, so there's lots of promise. We won't necessarily have a commercial product yet, maybe, but we'll make good progress towards that. And I agree. I think that you know, it's critical for large pharma to jump in. They already have started to do so, but increasingly. So we look forward to that. Great. So thank you very much. Great discussion. Great presentations. Appreciate it.
Thank you.